I think we're ready to start, so I would like to welcome all of you who have come. My apologies for being late. You know what is happening at Stellenbosch University. They are now searching for the new leaderships. I'm not in the running, but I, for some reason, was asked to be one of those individuals that give our opinions about leadership in higher education institutions in South Africa. And unfortunately, so I had to be on this Zoom. And the team that is putting together this report sits in Joburg. So it was either getting a bad rector next or being late for some 2 minutes. And I chose, for the fact that STIAS rents from Stellenbosch University this piece of land. We need some peace down there. But welcome, thank you very much, and we want to welcome you to the first public lecture in the series of 3 public lectures per semester. And thank you very much, Diethard, for accepting to be the first, for giving this public lecture. And usually with the public lectures, we do invite a colleague that is not part of the fellows to introduce the speaker. And we are glad that Professor Knox Makunga has agreed to introduce Diethard. But before I invite Knox, as we call her, I would like to make just a very quick announcements. For the public lecture, we ask the presenter to speak for about 40 minutes, not 40 hours. And then we have a Q&A, and then we have a glass of water after, so that I don't chase people that don't take wine after the process. So for the announcements, and since this is a public event, I'll only just announce the public events, STIAS public events that we expect during this semester. The first following public event will be on the 25th to 29th, and we'll be hosting the 6th International Youth Think Tank Youth Conference, which is also been upgraded to a Nobel Symposium, a Nobel Peace Symposium. So it will take place here from the 25th to the 29th, and some of the sessions within there will be open to the public. So we'll have about 24, 25 young people, leaders from the African continent, speaking about how peace can be achieved and also being the future leaders. And we've asked some of our fellows, including our Isolomso fellows, to be speakers during that conference / Nobel Peace Symposium. On the 26th of September, we'll have the public lecture by Godfrey Mwampembwa. So that will be the second one. And then on the 9th of October, we'll have a book launch titled A Seed of a Dream. And it's a book by Clive Glasser, who is a STIAS Fellow in 2018. I won't say that between him and his wife have also decided to contribute the STIAS Fellow this time. So we'll have a book launch. We try to encourage the fellows that have written their books or publications once they're done, if it's possible, to give us the honor to come back and launch their work. This may be after 1 year or 3 years or 5 years, or somehow, yeah. Then on the 21st to the 25th of October, there will be a Nobel Symposium in Physiology of Medicine on progress and challenges in cardiovascular medicine. And so we're looking forward. It will be the 4th of the Nobel Symposia that we've held. And I would like also to say that now, as I said before, the Nobel Foundation has agreed to fund another 6. So we do have symposia running into the 2030s, which is really an honor that we're receiving from both the Nobel Foundation and also the Swedish and Norwegian academies of sciences. On the 14th of November, there'll be the 3rd public lecture by the STIAS Fellow, and El Sabe will be from the University of Pretoria, will be speaking on aid flows to Africa. So those are the events that should be also on our website that are public, and we hope that we get public people from the public to come and be part of these just opportunities for us to show that we have individuals here and they are doing something. So Diethard is going to be the first one to convince the public that we're doing something at STIAS. May I now ask or introduce very briefly Knox, who is going to then introduce Diethard. Knox, as she's informally referred to, is a medicinal plant biologist in the Department of Botany and Zoology at Stellenbosch University, and her work is heavily invested in South Africa's renowned plant biodiversity that is highly resourced with plants that have medicinal values. Her work looks to identify the molecular and genetic regulations of the secondary metabolism in medicinal plants. In her laboratories at Stellenbosch University, the team has been applying a broad-based biotechnological approach to facilitate use of plants efficiently in a non-destructive, sustainable manner. So when she becomes a fellow next, you'll have to talk about that non-destructive, sustainable way of dealing with that. She has also acted as an honorary secretary, a vice president, and president of the South African Association of Botanists Council. And she also, I think most importantly, she serves on the STIAS Fellowship and Research Program Committee. So Knox, may you please come and introduce Diethard.
Thank you very much, Edward. Okay, so that people can actually see my face. It's really a wonderful and a great pleasure to have you here, Diethard, giving this public lecture here at Stellenbosch University. I think we really are in presence of a great mind. And I say this because Diethard actually has an extensive CV, and it is exceedingly impressive. Over 300 journal articles, many book reviews, and some book chapters. So it really is quite extensive. Apart from the science that he does, which is focused on evolutionary genetics, he also interacts quite a lot with different public engagements and has even appeared on television, from what I understand. And this career actually started with a PhD in 1983 in Tübingen, and that was followed with several different postdocs at the Department of Genetics in Cambridge in the 1980s, and another postdoc at the Max-Planck Institute in the late 1980s. And then thereafter, he then decided to carve his own niche and actually initiate his own research group. and that was around 1990 in the Department of Genetics in Munich. And this career then led him to continue in studying evolutionary genetics in various different organisms. And ultimately, he found himself as a member of the EMBO. And what I think is quite fascinating is that quite recently he was awarded the Federal Cross of Merit for his achievements in DNA fingerprinting. Now, this is really quite significant because many of us that are interested in evolutionary processes, evolutionary genetics, molecular biology, utilize the technique of DNA fingerprinting as a key technology that allows us to be able to unravel complex problems that might be related to life. And without much further ado, I'd like to welcome you here at STIAS so that we can learn a little bit more about the evolutionary fluidity of sex differences. And I think in relation to the recent context associated with some of the controversies at the Olympics, this topic is going to be of great interest. Over to you, Diethard.
Yes, thank you very much for this kind introduction and the opportunity to give this lecture, which is a great honor for me, and I hope I can live up to it. So, since you mentioned the controversy about the Olympics, I had actually this weekend I had a newspaper article in a Berlin newspaper about this topic, but it's in German, so I cannot forward it. So, yeah, you see, you had now time to think about where is Plön. Because every time I tell people I come from Plön, I have to explain where is it actually. So my first slide is usually about explaining where it is. So, so this is our institute. This is a little city where it is. So here is a map of Germany. You see Plön here is about 70 kilometers north of Hamburg. And, well, the city is in the Lake District, which is actually the reason why there's a research institute here, as you will see in the next slide. You may see this castle here. So this castle was actually the summer residence of the Danish kings until the Prussians took over the region. And then afterwards, it was actually the family of the last Kaiser, Kaiser Wilhelm, lived there. Actually, all the sons were educated at this place. So if history would have been different and we would have a constitutional monarchy nowadays, then this would be a royal place. Well, we know the history was quite different. Anyway, so you see here also the Max-Planck Society as a whole. So these are all the Max-Planck Institute across Germany, and the Max-Planck is like a big university, but at many different places. So it has humanities, it has biology and medicine, it has chemistry and physics, so it's really like a broad university. Total size is about like Harvard University, but our president always says, we have less money, but more Nobel Prizes. So this is the Institute aerial view, and recently, a few years ago, we actually had a 125 years anniversary, because this was founded already here in 1890, I can't see from 92, as a biological station at Plön, actually to do the research on the lakes here. This was at the same time when many marine stations were also founded, but then there was some people had the idea, why don't we also look at our own lakes? And so they looked for a place, and this was the perfect place. And that's why it is already an old research institute here. It was then, 1917, included in the Kaiser Wilhelm Society, which was the predecessor of the Max-Planck Society. All other Kaiser Wilhelm institutes were at that time in Berlin, only that was sort of outside of Berlin because of the location, of course. So it was therefore also became a founding member of the Max-Planck Society as the Max-Planck Institute for Limnology. So it started, did the ecology of the water surroundings there, and Some people say a substantial part of what you read in today's limnology textbooks is actually the research was done there. But then in 2007, it was changed into the Max-Planck Institute for Evolutionary Biology. New building activities started and are actually still ongoing there. Okay, now you know our institute and its history. Coming back to my topic, I found this by chance, this quote from Max Delbrück, because it gives a nice introduction. So Max Delbrück got a Nobel Prize in 1969, but that is a quote from 1949 in a little piece that he called "A Physicist Looks at Biology." He says, "The history of biology records discovery of great generality, like those of the occurrence of sexual reproduction in all living forms, and of the cellular structure of organisms." So he puts these two discoveries of the greatest generality that you can think when, as a physicist, about biology. And that's a wonderful introduction, because that's true. Sexual reproduction happens everywhere in life. Not all organisms do it, but the vast majority is doing it. So it gives a nice contrast to what I call evolutionary fluidity. So where are we getting at when we say great generality and evolutionary fluidity? And I hope this is the sort of contrast that I will be able to explain a little bit more today. But let's first go to the great generality. So we as humans had apparently the feeling we have to tell to people in outer space that we are two sexes. This is the famous plug of the Pioneer 10 spacecraft. That was the first spacecraft that was launched in a way so that eventually would lead out of the solar system, out of the planetary system, and go into outer space. That was launched in 1972. And Carl Sagan at that time said, why don't we take the chance to put a plug on it, so if aliens find it, they know where we are, where the Earth is, and that intelligent people have created this. So the plug includes actually a map here. These are quasars, so that you can actually locate the Earth, where it came from eventually. And it did locate, well, here you see also the spaceship itself as a size measure. And then you see humans depicted here as two sexes. So they could definitely have chosen to make only a sketch of one human, but they thought they have to be two sexes. So what did they want to convey? Presumably, we are a life form with a binary phenotype. But how would aliens see that? They could read it also differently. They could think, two different species have actually cooperated to build this spaceship. And there could be, some people say, there could be also another interpretation. It could say, well, we can build spaceships, but we do not know how to dress. So the important thing is they try to make a typical humans. And so one thing that is typical about humans, there's an average difference in human height. It's actually 8%, measured across very many populations. It's relatively constant, 8%. And it's properly depicted here. But again, if you say this, then we make a binary distinction. We say, okay, males, females, and there's 8% difference. But if you think about height differences, they are actually not binary. Human height is not binary. Because if you do this little experiment, this is college students put onto a field and sorted according to size classes, which are depicted here. Females have white dress, males have the black dresses here. And then if you look carefully, it's easier to see among the black, but also among the females. It's actually two different distributions, and they overlap by 32%. So it's actually not a binary phenotype. There's overlapping distributions, and we are all aware of it, and still we like to make binary distinctions between male and female. So that will be a key issue of my whole talk here. How did this come about? And now we go a little bit back into history. There are two versions of the mechanisms of heredity. So the first one, highly celebrated 2 years ago, Gregor Mendel, who discovered the Mendelian rules, as we call them. And he describes genetic heredity as a combination of independent entities. So here's a combination scheme of one locus and two alleles. But in fact, born in the same year as Mendel was Francis Galton. And he was actually, at that time, he was actually much more famous. He published this book about natural inheritance, and his topic was actually human height. And he tried to understand the genetics of human height. Naturally, he did not describe it as particulate characters, but he described it as a statistical principle. And so we have this confrontation between what is actually the nature of heredity. Is it a statistical principle, or is it made out of little pieces? Well, in terms of genetic sex differences, we can see it like this. So we have genetic sex determination by the X and the Y chromosomes, and they sort together such that you always get a 50-50 sex ratio, and that would be Mendelian. So Mendel didn't discover that, but that is a typically Mendelian inheritance. But the phenotype of the sex determination, of what makes the final body, is actually overlapping distributions, and that would be closer to what Galton had described. In fact, I asked artificial intelligence to make faces from beautiful woman to beautiful man and anything in between. Not sure whether it worked so well, but that's at least the idea you can imagine. There is always a range of phenotypes across both sexes, and there are persons who may be in between where you do not know exactly which sex they actually are. So there is a continuum as not a binary distinction. And only the very first decision here is a binary one, but then the later one is an emphasis of inheritance of continuous variation. Now, these are two opposing views. In fact, they were already in 1918. They were merged by the young Ronald Fisher. He worked out on the statistical link between Mendel and Galton, and his solution was probably, at least the biologists among you probably know it very well. So it's basically if you have a Mendelian trait with two genes, two alleles, one gene, two alleles, then you have three classes. If you have more genes, then you have more classes, and so on. And eventually you might approach a continuous variation if you have many genes. That's today's interpretation of these two forms of inheritance. So this is what I would like to call single gene genetics, and this is actually the most genetic work has remained here until today. So single gene genetics has really is the foundation of all of molecular biology. Knock out a single gene, find out its function, complement it, and show that the function is true. This is nowadays called polygenic genetics, because it takes into account many, many genes, thousands of genes sometimes, and many alleles, puts them together and treats them statistically. And this is actually only now being rediscovered to explain actually the heredity of the phenotype, because this does not explain the phenotype. Actually, my first weeks at STIAS were spent with writing a manuscript about this whole story, also about the history of how people were thinking about these different versions of genetics. I can send it to you if you're interested in it, but it's written for biologists, not so much for historians. Okay, coming back to sex differences. Keep this in mind, the polygenic genetics, because it will become important later. Let's go first to a classical question. The riddle of sexual phenotypes. So Darwin wrote already in 1860 in a letter to a friend, why should a bird like the peacock develop such an elaborate tail which seemed to be at best a hindrance in the struggle for existence? So Darwin thought about it very early, and the peacock's tail became a classic in evolutionary teaching. So here you have the peacock's tail, the male extremely exuberant display, and the female much more adjusted to the environmental conditions. So Darwin eventually, in his book in 1871, expanded on that. That is a book, is a follow-up book for his Origin of Species book. It's on the descent of man, but also on the question of sexual selection, a term that he coined. And he suggested that different selection regimes act on the male and the female phenotypes independent of the adaptation to the environment. So he said sexual selection is a second form of selection parallel to natural selection. So he set the scene basically, but since then we as evolutionary biologists keep struggling to find out what it really means. So consequences of sexual selection. The constraints on maintaining sexual dimorphism results in an evolutionary conflict for the corresponding traits. Now, what is an evolutionary conflict? So now you will see some cartoons in the way we usually present them as evolutionary biologists. So sex ratio is an evolutionary conflict situation. You may not have realized this because XY inheritance says already it makes 1-to-1, but in fact it's an evolutionary conflict because usually you have, well, most populations have a 1-to-1 ratio, but if you think about it, the females are the ones who bear the offspring, and it should make sense to have more females than males because a few males can inseminate many females. So why do you have all these superfluous males? Okay, so a mother could think, it's better to have more daughters. Now, of course, a mother doesn't think that. That's how we present this usually. But there are genetic drive elements that can do exactly that. So what will happen? If we have more daughters, then we shift, they have more females, less males, and then an interesting effect kicks in because then the relative fitness of the sons increases since each will have a higher fraction of offspring. So the value of a male is higher than a value of a female in this asymmetry. So what happens? Now we need more sons. So now you can shift again back to the other situation. But of course, in this situation, what happens? The relative fitness of sons decreases because each will have a lower fraction of offspring. This is currently the situation in China, presumably. So there is a problem then. So what's the solution? Well, grandma tells us the solution, stick with 1-to-1. So 1-to-1 is what we call an evolutionary stable solution, ESS called. And the ESS is evolutionary stable, but means there is active evolution going on all the time. It's switching here, switching there, and goes around 1-to-1. But it means in the background there's a lot of evolution ongoing. This may remind you of those who have heard Dieter Ebert's talk on the Red Queen model. It's very similar. You have to keep going just to stay at the place where you are. And the Red Queen model is also an example of evolutionary conflict, in this case of hosts and parasites. Okay, so this is evolutionary conflict. This is one of the consequences that we have under sexual selection. And the other is that we have different trait optima. So different evolutionary interests of males and females lead to different trait optima. We result in sexual dimorphism. So what are trait optima? So again, depicted as a scheme. So we have here fitness curves, and males may have a different fitness curve than females. Now, as I'm going to talk about genes, we kick now in what we call expression level as a measure of gene activity. So under these conditions, if you have different trait optima and you have an expression level that is the same in males and females, then it's suboptimal for both of them. So what will happen is that you will, for genes that are relevant to the sexual phenotype, you will develop different expression levels. So you have a difference between males and females. So the gene activity will be adjusted to the sex differences. And now that's a great possibility to go into the genome and ask what is actually the expression level differences between the different genes, and then to pinpoint the genes that are relevant for sexual development. That's at least the theory. just going back quickly for those who are not so acquainted with the molecular biology behind it. First of all, the term is here sex-biased gene expression. And I think this is intuitively clear. There's a sex bias here. Well, most genes are expressed the same in both sexes. Now, why do we use gene expression as a measure for gene activity? So here's just a sketch of how genes are transcribed from DNA and the process, and then in the cytoplasm become protein. And we focus here on this part, so the messenger RNA level, which is what we call expression level. So why are we doing that? Probably it would make more sense to look at the proteins rather than RNA. Well, we're doing it because we have the machines to measure that properly. So we have these little helpers which can measure 1 billion such messenger RNA, or count 1 billion messenger RNA molecules per day, basically. So it's, you get a huge statistical basis for measuring the gene activity. Okay, so this is the experimental regime that we used to a large degree, and this is where I want to tell you some data. We use the mouse system to do that. So that's the house mouse, and we have developed this in the past years. We have collected mice from all over the habitats where they occur naturally. It's not important for this talk where they come from, just to give you a feeling. More important is that they have a very defined evolutionary relationships. And that's a good setting where you can actually study the evolution of sex-biased gene expression, because they are relatively closely related. So you have these two major branches, 0.5 million years. This is 2 million years. Now, 0.5 million years may be, well, I don't know whether you can imagine what 0.5 million years means in terms of evolution. So, for example, we as Homo sapiens are about 0.25 million years old, but our separation from chimpanzees is about 5 million years. So, but because mouse has about 10-fold more generations per year, the evolutionary distance, 0.5 million years in mouse, is approximately what is human chimp. So that you can keep this in mind. Relatively shallow still. Okay, so we use 4 populations that I indicate here, and you will see these Domus, Spretos, Spitzelegos. You will see this in several graphics here. It's always in the same order I present them. And then, of course, now the next thing is when you look for sex-biased gene expression, you have to decide which organs do you look at. Of course, you can use the whole individual, but then you mix up gonadal organs with non-gonadal organs. So we decided to make a separation. So we used 5 somatic organs, so brain, heart, kidney, liver, mammary glands, and then sex organs, ovary, oviduct, uterus, testis, epididymis, and vas deferens. Okay, so that's the setting. Now, this is only for the specialists, so there is statistics how to do that. We take the ratio of the female-male transcript counts, make a cutoff, and then use also a statistical test. But not important for the further talk. All it's supposed to do is tell you, yes, there's a lot of statistic and has taken a hell lot of time to work this out. But now let's go to the results. And I usually start with this slide, because now I let you guess which tissue has actually the largest average number of sex-biased genes per somatic organ. So those tissues which must be particularly important for male-female differences. So I give you the choice between brain, heart, kidney, liver, mammary. Now it's up to you. What is the most important tissue for sex-biased difference? Anyone wants to have a guess? Memory is a good option, yes. Oh, I should have said, in mouse, the mammary glands between males and females are actually identical before a mouse before the mouse becomes pregnant. Okay? Brain, yeah. It's amazing how many people say brain. brain has the least number. So this is the average across all 4 species. So brain in all 4 has very few. Heart has more. Actually, the internal organs, kidney and liver, have a very high number. And the memory is also among them, but actually kidney is higher. So that's already interesting, let's say. But now this is an average. Now I present a big data slide. I'm sorry for it, but I will go through it, okay? Because now we look into the 4 different species, because now it becomes interesting if we do that. So you see here all the organs. So these are the somatic organs: brain, heart, kidney, liver, memory. Here the sex organ comparisons. Here again the tree, so that you can orient yourself in the 4 species. Now let's start with the brain. As I said already, in comparison to all others, it's actually the lowest number. Then the heart, I showed you, was sort of intermediate, but interestingly, if you now go to the details, the domesticus here have a huge number of sex-biased genes in females. This is the outgroup, so they must have developed it only very recently. And that gives you already the feeling this is not a very stable system. That changes very fast. And you see similar indications actually in the other organs, where they have, between these relatively closely related species, major changes in the numbers of sex-biased genes. This is not the case if you look for the sex organs. For the sex organs, you have relatively stable situations. So it tells us already that the phenotype of the sex is actually something very fluid, apparently, in terms at this level. We come also to other levels. Now, this is actually one of the, as I explained to you, predictions of the evolutionary conflict, that the genes that are behind it should evolve very fast. And you got already a feeling from the number of genes, how quick they change, but we can now look also in each individual gene. And I do this here first for one gene. So this is a gene with a conserved female biased expression. This is a series of individuals from the 4 species, always a group of females, a group of males. Red is female expression, blue is male expression. Because this is female biased, in this case it's actually purely female expression, no expression in males at all, okay? Now let's go to a gene with a non-conserved female biased expression. So here again, here you see a similar pattern as here, but here in those you see equal level of female and male expression. So the bars are the expression level in each individual. And then you can have also this situation. You can have a female biased here, non-biased here, but male biased here, within a spread of 2 million years. Any particular gene can change its role, apparently, in this conflict of sex bias. Now what we can do is now ask the percent evolutionary changes just in these mouse species in the sex-biased gene. So which percent is conserved across all of them, which percent is changed, which percent is reversed. And surprisingly, only 3% are conserved in sex-biased expression. 95% change their role in any of these species within a 2 million years distance. And 2% actually switch their role. So there is a tremendous evolution at least at the expression level. The genes themselves, of course, are there, but they change their relative expression level. Now, I showed you these overlapping phenotypic distributions for sex in sex. Is it possible to find overlapping distributions also in the sex-biased gene activity? So currently I showed you only single-level expression, but you may have noticed, as a sketch from the previous slide, that each female has actually a different height here. There is actually quite some variability between the individuals, both in the females and the males. And you could imagine, so there are some males which have the same expression as the females here, for example. So how do we combine this into a single big factor to make it visible like these overlapping distributions? So we have developed for this what we call a cumulative sex bias index, call it SBI. So the logic of that is, as I introduced, so the sex bias gene expression conveys the sex-specific phenotypes. So all genes that are sex biased should somehow contribute to the genotype. And then we say, okay, why don't we just use, in this case, the median expression of all sex biased genes in an individual? And this is then a measure for femaleness or maleness. Now, I underlined the all here because that is actually a deviation from the classic genetics where you go gene by gene and ask for each gene what is it doing. This is actually now in the new thinking of what we call polygenic genetics, where we think it's actually very many genes contribute to a phenotype, and we use a kind of additive measure of them. We don't know exactly whether it's additive, but as in first approximation, it is additive. So these indexes are now being developed also by breeders, for example. They make a breeding index value across the whole genome. And there is also now a disease incidence index in medicine and so on. So we introduce it basically as a sex bias index. So how do we calculate it? So it is relatively easy. So the sex bias index is the median expression of all female bias genes minus all male bias genes. So so put them together in a single factor. You have to put them in relation. You could also do, of course, male bias minus female bias, but it doesn't matter. So here it's a number of individuals, and if it in the SBI, if you're a female, then you have, of course, more female biased genes, and then you go up here, you have positive values. Males, you have negative values. So you get a distribution of a single factor, and then we can put this actually now into distribution plots. So these are the sex bias index plots, and now you see in this hypothetical example, you have already an overlap between the distributions. Talking of hypothetical examples, what could we potentially see in such a sex bias index? We could see binary patterns, so extreme differentiation or strong differentiation, or there could be non-binary patterns, meaning there's overlap, either partial overlap or almost complete overlap. So this is what we can now distinguish and ask, how do we have mostly binary or do we have overlapping pattern, whatever. Just to remind you, so this would be with the human height phenotypes. There would be partially overlapping, as I showed you with 32%. But this is now, of course, gene expression and not a measure of phenotype. Well, it's another measure of phenotype. Now, what are the real data look like? So here I put out only a subset of them. You know, with the many species, many organs, we could do many combinations, but just to show the point, so we have, I use the domesticus and the Spratus as a comparison, so they're 2 million years apart. And these organ comparisons, here the brain, not much changes between them, but the heart, we have here a large overlaps. For the liver, we have here a clear binary pattern in domesticus, but an overlapping pattern in Spratus. Again, something very surprising, actually for us also, until we finally read the literature and found, yeah, people knew already that the liver in mice is sexually extremely distinct between them, physiologically also very distinct. So if you recall the earlier slide where I showed the numbers of genes, these are the panels from this earlier slide. I just point out this one. You recall the many female biased gene in the mouse, in the domesticus, Now, if you look now at the sex bias pattern, so they have many genes, but they overlap actually. So it's actually, many genes alone do not tell you there is an extreme sexual differentiation. The overlap of the sex bias is actually very high. Here you have also a decent number, but then the sex bias index makes them very different. So it gives you two different types of information, the number of genes versus the sex bias gene. Now here for control, of course, the sex organs are extremely separated in this sex bias index. Okay, that's for the mouse. Now, of course, now we can go on and do the same for human tissues. So we use the GTEx portal for that. So this is a collection of many tissues from people who donated their body to medicine and then agreed that they could be sequenced and that data could be provided. Of course, they're all anonymous, but one can use the data publicly. And the advantage is there are lots of tissues. I have to make such a list. I'm sorry about this. Starting with sex organs, ovary versus testis, then the mammary tissue, breast, fat tissues, and so on. Now, if we use the same statistic as for the mouse, asking first how many genes are there, it turns out, and I ordered them already in this way, so the largest number is in the sex organs differences, but then here we have actually an extreme difference in mammary tissue, or extreme numbers in mammary tissue, because these are adult, they are anonymous, so we don't know whether they had children or not, but probably it doesn't matter in humans. And then there's another tissue which is extreme large numbers of sex bias genes, is subcutaneous fat tissue, so it's the fat below the skin. There's a second fat tissue which is called visceral omentum, which is lining the organs, and which is known to be actually physiologically different. And it's only the one under the skin which has an extreme number of sex bias genes. So in this list, the organ which is most different in humans between males and females is the fat tissue. And in retrospect, it seems to make sense, because this is indeed what also makes our shapes different and so on. Actually, also the mammary tissue is, of course, mostly fat tissue, so this overlaps a little bit here. Then again, blood vessels are slightly different. But you will also see that in many tissues, there's actually very few female sex bias genes, actually exactly one. And this is a functionally necessary one that is required to silence one of the X chromosomes. So compared to mouse, we as humans are much less sexually differentiated. Yeah. Now, most tissues have very low numbers, especially few female bias genes. And then the sex bias index across these tissue. Of course, I cannot do an index when only one gene is expressed, then it doesn't make sense. But so it's only for those tissues which have multiple genes. But you will see, without going into much detail, they are all overlapping. Even the adipose here, where we have lots of genes, but there is overlapping distribution, but less variance in females than in males. So only ovary and testes are well separated. So overlapping distributions here in humans as well. So we have no binary sex phenotypes at the transcriptome level. So I'm slowly coming to the end. So now I've contrasted here what we could call the duality of the sexes. We have the binary switches at the chromosome level. And molecularly speaking, this is indeed the only binary switch in the development, where only a single gene switches from a female pathway into a male pathway when it is active. It's actually a gene from the X chromosome. But then all further developments of the phenotype are actually polygenic. And then you have actually distribution of phenotypes. They are not binary anymore. So you have overlapping distributions in particularly. So based on these considerations, I have actually published also in the TV that Knox mentioned. I've said we should give up registered males and females anyway, because it doesn't make any sense. So because the law tells us we are all to be treated the same, so why does the state register the sex of people? If you think about it, it doesn't make any sense. So why do you have to put into your birth register some decision about your sex? You are actually part of a continuum, and it's up to you to define yourself later on. The state doesn't need to know. I know it's a bit radical, but who knows? Think about it, I think it makes sense. Anyway, so you may have noticed that I have carefully avoided one word so far, which seems to be unavoidable when you talk about sex. It's gender. So I want my last 2 slides I want to spend on this discussion. Here I have a sketch from 2019 in the Lancet, which tries to explain people what is gender, how do we understand gender nowadays. And of course, they make the binary distinction and they put here into the say sex is biological. So it's the chromosomes, reproductive organs, hormones, but all the rest about is actually culturally defined. Now, this is, of course, how people like to think about this nowadays. It's part of our culture. Well, not everybody, but I think many people will agree that there's a lot of cultural influence of how we act as the sexes. I want to contrast this with a quote from Darwin, which is one of the quotes which those who like Darwin do not like to put up usually. Because Darwin lived in a completely different culture. So Darwin says, the chief distinction in the intellectual powers of the 2 sexes is shown by man's attaining a higher eminence in whatever he takes up than can women, whether requiring deep thought, reason, imagination, merely the use of the senses and the hand. So this is from a critical, independent thinker, has completely reflected the environment, the socio-cultural environment of the Victorian age. I think Victoria was still queen there, so he probably insulted the queen by then. But it's very difficult to say. But this is even these independent thinkers are part of their culture condition. And I think the same is actually here. So we have currently this swing back into everything is cultural and the biology of the sex is only the nitty-gritty at the basis. So my argument, and I hope I could get this over, it's all overlapping. So we cannot say what is culture, what is genetic. There are definitively, if you have these overlapping distributions, it means there are also some females or some males which are typical females and typical males. It's just you have overlapping distribution for each individual. For some individual, it might be impossible to tell, but it's not all cultural. There is also some genetics behind it and it gets remixed all the time. And that's now my final slide, coming back to this again. So I'm now added something to this. I say, okay, the great generality is a fast evolution of the elements of sexual reproduction. So sexual production is everywhere, but it evolves also very fast. And that's, of course, a conclusion which has been taken by many evolutionary biologists before me, probably already Darwin. Okay, so thank you very much. And that's where I want to finish.
Thank you very much, Diethard. And I think you've, in a way, also helped the National Statistical Department to not, take time to say male, female, or something like that. So we would have to set up a meeting in Pretoria and see how the next population census is going to work. But thank you. I would like to recognize all of the colleagues that have joined us online. Unfortunately, I can't see them, but they're all most welcome. We would like also to welcome, I can see our fellow from last semester, Hans. Thank you for coming, and quite a number of colleagues coming from the broader Stellenbosch area. I see one lady who is very keen in public lectures at STIAS, sitting over there behind Alan. She always comes for these public lectures. So on that note, I would like to open the discussion, the conversation, to engage with these overlapping genes and the conversation that again goes into the complexity of us and engage Diethard in this conversation. Yes, we start with.
Thank you, Diethard. I would like to ask you about the interpretation of the sex bias index. Would you mind going back to one of the slides that is entitled Evolutionary SBI Comparisons? Okay, so you have emphasized a lot the difference between a binary, strongly pronounced segregation in these two distributions versus an overlap situation.
Right.
But I'm missing two things. I'm missing the interpretation of how far the values are apart in several cases, because there may be segregation of the two peaks, but the two peaks may be closer in value than in other situations where there is a large overlap. And also I'm missing the interpretation of the absolute values, because in some cases both peaks are on the positive values, and in other cases both peaks are on the negative values. So how does that relate to male-like or female-like?
Yeah, you have very sharp eyes. I had hoped that nobody would notice that. So the paper that we have written on this was just reviewed, and one of the reviewers also picked on this part. For us, it was implicit because we are so much used. The x-axis has no particular meaning. It has only a complete of the absolute value of the median, but this is not what we are trying to compare. We are only trying to compare the overlap. So the scale is actually the same in all cases, if you will notice. But the relative position along the x-axis, and that's a property of this, how we calculate it, is not important. So I will make a different, so we should actually remove the numbers here to avoid the confusion. So the relative position on the x-axis is actually not relevant for what I'm saying. It's really only, you should see the distribution. So if you have here such a broad distribution, it means there's a lot of variability among the individuals. If you have a narrow one, this is very little variability of the individuals. This is what you can see from here. Sorry that, I should make a new version of that.
Yeah, that was mighty interesting, of course, and maybe anti-controversial nowadays. But I was thinking the origin. One often discusses nature versus nurture, and for me, nature is genetics. I'm not a geneticist; I may be wrong. And nurture is the other stuff, environment. You looked at RNA, and therefore not at genetics only, but substantially, potentially, at nurture. How would you reckon it? Has there been a way for you to see how much is nature here and how much is nurture here?
Well, that's one of the insights of this polygenic genetics is actually that no gene system is independent of the environmental conditions. So if you put the same gene system, so there's always a term in polygenic genetics, this is the genetical part and this is the environmental part. So for example, for human height, which is the very best studied phenotype by now, it's estimated to be about 50% is genetic and 50% environmental variables. And you can even there are studies, I know one study from Colombia where they looked at height measures recorded by the Statistical Bureau, and then could actually, so the average height of the people becomes lower when socio-cultural conditions are bad, and when they are good, then they become higher. They could directly follow this in the statistics. So yes, so all of these polygenic systems are a combination of environment and not necessarily nurture. Environment could be lots of things, could be epigenetics, could be, could be temperature, could be food, could be whatever. And these combinations we don't understand yet. So we really have to do much more research to better understand the interaction between environment and genetics. So we cannot completely disentangle it. You can do it in certain quantitative experiments. We cannot do it here. Surely the expression levels, also the variation of the expression levels, will have an environmental influence, particularly in humans. And that may be the reason why they are more closer together, because there's more variance in them. The mouse, although these are outbred mice, so they're genetically variable, they were kept under constant conditions. So the environment was a bit reduced. So definitely it has an influence, and the play between environment and genetic, we don't know exactly, but this is part of the polygenic theory nowadays that is developing.
Yeah, we have data here, and I would like also to recognize Florian, former fellow, and I'm waiting for the mathematical question. So, please, Diethard.
Thank you, Diethard. This was very inspiring. I'm trying to think more about the underlying basis. So in the end, what we also expect when we have two sexes is that they both fulfill their biological function, which the primary definition of males and females would be then that the female sex is the one producing the larger gametes and the male one is the one producing the smaller gametes. And then you have various consequences of this. If I now think about how do I define sex, I could go and look at the chromosomes, and then I find XY and XX, and then I say, okay, male, female. I could go and look at the gamete size, and I find the big one and the small one. Or I can look at any other trait, and then it becomes fuzzy. So where do we put biological function in here related to sex? Because what you are doing here is blue, you define, like people did this in former times, the male babies get the light blue pyjama and the female babies get the pink pyjama. So you classify them at birth and then you put your graph on it. But wouldn't the proper classification be to do it by function? And then how does this relate to this variation?
Yes, you can imagine that I had this discussion already very often, particularly with biologists. Biologists are the last ones standing who want binary sexes because, well, there are two types of gametes, they're asymmetric, and that determines all the rest. So this is often the argument. Now, you know equally well, and every biologist knows equally well, that the function of sex is to create diversity. Sex is used for recombination and shuffling the gene pool. So why are the biologists so preoccupied that the function of sex is reproduction? It's actually to create diversity. Reproduction you can do with other means, but the diversity is the important part. And that's what we have to see. And then if you come from that side, then it's obviously that the diversity can have overlapping distributions. Now, the biology, you have to see the ontogeny of the animals or the humans. And that's why I emphasize the only binary distinction is at the point where a gene from the Y chromosome switches female development into male development. This is what we can pin down usually onto a single gene. But then all the following already becomes diversified because the zygote that is developing, or the embryo at that time already that is developing, has of course a reshuffled genome already. And then all the phenotypes are polygenic that they derive from that. And then of course you get a body that is usually can have 6 different phenotypes, which we then can use to sort the animals according to these original classes. But then, for example, in humans, there have been endless numbers of papers who looked for differences in the human brain between males and females and couldn't come up with anyone. So already at the brain, you have already equilibrated the original binary switch. And now it's a question of, well, where do we want to look? Do we want to look at the gametes? Do we want to look at early development? Do we want to look at the individuals that interact with the environment? And that, I should have said, is actually my main project here at STIAS, to have a new look on the individual in the background of polygenic genetics, of what it really means also for evolutionary theory. If we look from the individual that interacts with the environment, then a binary description is not appropriate anymore. But of course, yeah, we had to get these data, we had to sort them original into males or females, otherwise we cannot do the comparisons. But then we are interested in the phenotypes, and the phenotypes are at a later stage. And that's where we do the transcriptomics.
Yeah, thank you for a really interesting talk. I'm just trying to wrap my mind around this fast evolution. What are you exactly referring to with fast evolution here? I mean, if you look at the transcriptomic level, we know that relatively few changes can completely change transcriptomic readings throughout organisms. Is this a fast evolution, or is this just a playground where you can score differences?
Yes, so that's indeed, we struggle still with the right comparisons. If you come from transcriptomics, you know we can do an overall divergence of the transcriptome or the regulatory divergence. And in this phylogeny, we get, for the non-sex biased genes, we get about half of them change their regulation in this 2 million years. Here we find 95% of them change. So it seems to be faster. But it's not a completely fair comparison because there are different mechanisms at play, and then you don't know. So we struggle with what is our comparison. The most important is actually, and I left out the slide, is when you now ask what is compared between mouse and humans. What is conserved between them in the end. So, you know, mouse is a biomedical model system for humans, and many genes, the disease genes, are tested in the mouse, and people think, yeah, there must be some conservation. Among the sex-biased genes, it's a very small handful that is conserved. Actually, interestingly, these are histone methylation genes, so epigenetic modifiers, which occur in pairs on the X and the Y chromosome. There's probably another evolutionary conflict going on there. These are the ones which are conserved. But the vast majority is not conserved. And this is, for me, the best way to say this is a fast evolution. So if you think about gender-specific medicine, you cannot use the mouse as a model because there's a completely different set of genes to achieve the same goal, but it will be different genes. So that's my main argument, but again, I mentioned already, of course, was another comment from a reviewer, we have to quantify or we have to set a scale, what is actually the expectation. It's very difficult to do that, but we work on that, we have some ideas, yeah. But the main argument is there's practically nothing is conserved. So one is of course cyst, is the one which is involved in X chromosome inactivation, but otherwise, almost nothing.
Any questions? Yes. So let's go here, and then we'll go to.
Hi. So, I mean, it was really striking to me in the mouse that most of these genes seem to be exhibiting female sex-biased expression, with very few being overexpressed in males relative to females, whereas that seems to change in humans from that quick table you showed. What are the reasons for that?
We only will know. Well, what was the answer to that? So the one answer is we are probably underestimating the number of genes in humans, because of the higher variability. So because we have statistical cutoff, so if you have high variability, then it's much more difficult to get a significant difference. So we probably underestimated, but probably not by a super large margin. So there is some noticeable shift. So for whatever reason, the mice, all 4 species, have usually more female bias genes, and in humans, there's a tendency to have more male bias genes. But I'm sorry, I have no other explanation that I could offer at the moment.
Mike has already moved. Yeah.
Forgive what may be an ignorant question, but I had a question about the underlying data for human populations. That you mentioned this is a tissue from a database of people who have left their bodies to medical science. But surely this is only a very particular snapshot of human populations. It's people in certain parts of the world who have access to technologies and medical facilities who are able to leave their bodies in this manner. How do you control for that?
Well, it's collected in New York mostly, or on the East Coast in the US. So it's probably takes into account different races, but they won't give you the data for them. So this is part of being private data. The only data we have is the sex of the individual and the age of the individual. And then the death reason. Was it an accident, or did they have a disease, but they wouldn't tell us which disease. So that's the only thing we know. So of course the creators of the data set, they took care of that, but in which way they can hide between the necessity to keep them secret. So we don't know exactly. It will not be super representative, but one other thing that we always should keep in mind, that humans as a whole are one of the youngest species on Earth. So, so as I said already, we have 250,000 years of history of Homo sapiens. That makes about 10,000 generations. So all of us are not more than 10,000 generations apart. So we are actually, in many ways, very similar. Although we know how to emphasize the differences. We know there are black people and there are people with a pigmentation deficit. But that's only superficial. So much of our physiology is probably not so super different because we are all one species and a very young one.
Right. Let's see, light.
Thank you very much, Dieter, for this great presentation. Okay, I have one question which is a little bit stupid, but I'm going to ask it anyway. When you ask us, when you present us the different organs and ask us which are, or which is the organ that makes the huge difference, we were all assuming, I mean not all, but most of us were assuming that it is the brain. And you say, oh, you're wrong. Could you tell us where does these wrong assumptions come from?
Yes, let's say when we saw the data first, I thought that's a great question to ask to the audience because somehow I would have intuitively also thought the brain must be different because, yeah, I mean, it goes back to Darwin. Darwin said the brain must be different between males and females. I don't know. It's an interesting preconceived thing, yeah. And it works in almost every audience.
Shall we then have Yoki?
Thank you for the presentation. So as a gender scholar, I think this makes us very happy because this is what we've been saying for a long time, non-binary thinking about gender, and now looking at gender and also sex as fluid, sex for intersex, of course, and gender including now transgender people. And so I'm curious, are you having any conversations with gender people? Or because this is, I think, would give so much backing to some of the conversations going on. And also something else, maybe just a comment. many African societies have been seen, especially in terms of gender, not sex, to be fluid. The gender is fluid, and you'll find people move from, being a man, especially, half the time people move from being a woman to be a man, a social man, because there's a lot of power, of course, in becoming a man. So also, how does that speak to your work? And I'm also curious about the article you wrote about the Olympics and the debates with intersex.
So of course that is behind all that, but I'm a biologist, so I would say there is an interesting effect. As a biologist, we are allowed to talk about every living organism on Earth, but as soon as we talk about humans, we hit a glass ceiling. We better not should be asked as biologists, particularly about all the socio-cultural discussion about transgender fluidity. I think what I wanted to say here is, of course, this is implicit. If you say there are overlapping distributions, those people who find themselves in part of the middle of these distributions may not be able to say exactly what are they. They are not binary. They are somewhere, and they might even be fluidic. But this is really something, that's going too far beyond my data. All I want to say that the binary distinction doesn't make any particular sense. It's just there is all things in between. When you say, are we talking to transgender people? Sure, we have in our institute a good diversity of people with whom we talk, and also some sex reversals, and so of course, so we live with them nowadays. So we know them, we know the people. But of course, the discussion among them is much more heated, and there's some development which I do not like to say. Okay, now we create additional classes. I'm the class of whatever. I don't even know how they call themselves, but there are 5 different classes into which they want to organize and say, I want to be recognized as a class. And I think that's the wrong way around. I think we should actually remove the distinctions and say, we are all one humanity with many different phenotypes. Yeah. But of course, I know perfectly well the discussion here is much more complex. Yeah. I'm aware of the complexity of discussion, but I do not think I can very productively contribute to it. But sorry? Oh, the Olympics. Well, there was this amazing claim that these 2 boxers were males, and they didn't even specify what they did. They may have done chromosome tests, but it's entirely possible to have a Y chromosome and still be female, because, as I said, there's only one gene on the Y chromosome which has to do one switch early in development. But if this gene has a defect, then you have a Y chromosome, but you're still a perfect female. And that could happen with these. But I don't know. We don't know any details. But the argument that I made eventually is a similar one, as we're saying, why do we distinguish? So sex is one of the genetic differences that we have. There's no doubt about that. But it matters only for sports level at the Olympics level. There, of course, so if you have 100-meter runners, so the world record for females is around 10.5 seconds, for males is about 9.8 seconds. Now, 10.5 seconds, how many men will ever reach 10.5 seconds? I mean, these are extremes of the distribution, of course, these super sportsmen. They are extremes of the genetic distribution. And then it becomes relevant if you have the extra advantage of being male and having more muscles and whatever. And there it makes sense to make a distinction. But I think for the average sports people in the middle of the distribution, it actually doesn't make sense to differentiate it by males and females. You should differentiate it by their by classes of what they can achieve, which is my average run time, and those with a similar average then should compete each other, because at that level the training makes a difference. That's a fair competition. The sex is relatively unimportant at that stage. So that was my main argument that I made. For the case of the boxers, of course, I have no practical insights. I only recommended one should make a decision for a certain person at one point and then not question it again. This is always in sports. You have to make decisions about inclusion or not inclusion. This is a female, this is not a male. And if it's once decided, then one cannot question it over and over again. So, yeah.
I do see a hand right. Still, the hand is still up.
So, as a biologist, if I want to classify, let's say, insects, and I want to look at 2 different bee species, I'll probably find that the majority of their traits are overlapping. But if I look at a small handful of traits, I'll find differences between them, and I'll be able to classify them as species X and species Y. And the point is that I don't look at all of the traits. I look at a couple of diagnostic traits, and I feel that it's worthwhile separating different species. And similarly for humans, I don't want to look at all the traits in a human's body if I want to classify a human as male or female. I only want to look at a certain trait or a couple of traits, and for the most part, those are extremely binary. And so I'm not really sure where to go from there.
Yeah, but you take as an starting assumption that classification is the aim. So if you want to build up a classification, which is perfectly valid for taxonomists and people who want to differentiate species and want to study the role of the species in their environment, then you need a classification to recognize them again. But within the same species, I mean, that's also a nice quote from Darwin, is that the differences of the sexes, phenotype of the sexes is sometimes so large that you would put them into a different species or even a different genera. When you know they belong to the same species, then you have to ask yourself, why do I want to make a classification? What's the aim of my classification? What is the added value of making a classification? It's always possible to classify, and our brain is so extremely tuned to wanting to make always classifications. But the reality, that's the interesting thing. Your brain makes the classification, but then you look around us, and there's nobody exactly fits this classification. They're all different anyway. So, but you want the classification, and then you have to say, why do I want it? And that's why I bring up the state. Why does the state want to have a classification, male and female? And you will find out that the state has no reason. At least the modern states that say everybody is equal.
I think as a sexually reproducing organism, we're kind of bound to make those classifications, because if you can't make those classifications correctly, you're basically going to go extinct. Your lineage will go extinct because you can't find the right person to reproduce with.
Well, I think, yeah, that's part of our evolutionary success, that we do not have problems with finding the mating partners when we think we want one. And yeah, but that's not what I mean with scientific classification. If it's a scientific or administrative classification, of course, males and females have to find each other, and there are many ways how they can do that. And it can be many different triggers that actually make people mate in the end, so it doesn't have to be a single trigger. Particularly also in mice. In mice, there's a lot of scents are very important for successful mating. So we have lost these feelings, and we don't even know what they feel about their mates.
I think our evolutionary succession is based on the fact that we are good at making those classifications. We make them all the time, and they work 99% of the time.
No, we are optimized to make classifications. We have to make classification. We have always dichotomous decisions, often, for example, in terms of aggressive or not aggressive interaction between individuals. So our brain is made to make this dichotomous distinction, but the reality around is not dichotomous. So that's actually also part of my project here, to get this sorted out from each other.
Great. Karina will give us the last question.
So you know that for humanities or a social scientist is asking a question when they're prefacing it by this is probably a stupid question. So having outed myself, you are very careful not to make pronouncements in the domain of culture, politics, gender. I detect there's a limit which you say you can't really speak to that meaningfully, and that there's a lot of nuance. So I wonder if we, for a moment, then take away that world of gender politics, the historical record of discrimination, etc., what are the stakes in your own field related to this kind of argument for fluidity? Or is it really the case that the stakes for this kind of argument sit very much in the domain of culture, politics, gender, and a longer historical understanding?
One of these deep questions that I have actually started to try to understand this myself. Where is the origin of... so if biologists talk about humanities, then you always hear very quickly the word biologism. So you argue from a biologism perspective. We are not going to accept biologism. So where did this come from? And my current research, actually also done here in STIAS, I start to read philosophical literature now where I have the time. It's probably going back to Kant, because Kant was the first to say the moral of the people should be independent of any scientific foundation. It's a moral in itself. It should not depend on the zoology or biology or any other inferences. So he made a separation. And I think the humanities see themselves also in this realm. The moral of humans is something which you may claim to have a biological root, but it's treated in a different way, I think. Now, interestingly, I also understand that Kant is nowadays not very popular among the modern thinkers in humanities anymore. So maybe even that distinction falls down. And then, but then, what do you replace it with? Do you replace it then again with biological arguments? And then, of course, you get an asymmetry. Then you have the biologists who are extremely in their subject, and the humanists who will have difficulties to follow all the arguments of biologists. And then, where's the discussion going? It's an interesting, very basic question. I'm happy to continue the discussion for ages. There's probably no solution, but yeah, we have to be aware of this. Yeah.
Well, thank you. I think, again, ending on Karina's and probably linking with some of the questions is to see, Diethard's work, how it links to the perceptions that are sort of helping us to jump to conclusions. And the point that you made about, how the work now can actually be used to interrogate the Kantian disease, as they call it sometimes, and then to say what philosophical standpoint now kind of becomes the basis for the arguments. And I think also the importance of bringing the biologists. You're very careful to say you were there, but now in STIAS, you're being brought into the pot of the biologists, the philosophers, the historians, and it'll be interesting to see how that rubs off the biologists. And so we're looking forward to that, and I hope that you can also be invited to speak to the sociology, gender department. I'll escort you so that the sociologists don't eat you alive with their aspects. On that note, I would like to thank Diethard for the presentation. Thank you very much, and please give a round of applause to Diethard. Genetics, we can move on to have a glass of water and continue the conversation.